There we go. Yep, and so Russ has been sort of contributing in a lot of different areas. He's currently at Stanford, and he's the Albert Raylang Professor in the Department of Psychology, and he has appointment also in computer science, and he directs the Stanford Center for Reproducible Neuroscience, and he's been focusing on the use of neuroimaging to understand decision making and executive function. And he's also been spending a lot of effort in the reproducible neuroscience area, focusing on neuroinformatics tools, increasing transparency, sharing of data, and cognitive atlas ontology, which he'll probably refer to today in his talk. So without further ado, I will pass it over to you, Russ, and thank you very much for taking the time to join us today. So
sure, thanks for the invitation. Are you seeing my slides now?
Yep, looks great.
Cool. All right. So yeah, thanks for having me. I want to talk about open and reproducible science, and I want to start by basically saying, what do we mean by, in particular, reproducibility? I think most of us kind of know what we mean by open. This is a nice slide from the Turing Way, which is actually a great resource for reproducible science, that kind of highlights some different aspects of the different ways to think about reproducibility, right? Which really had to do with, like, are you talking about the same data or different data? And then are you talking about the same analysis versus the different analysis? There's various versions of this. I'm going to stick with this particular terminology today. And so what I'm going to do is kind of walk through each of the quadrants of this figure and ask, how are we doing in that quadrant? So first, let's ask, are neuroimaging results generalizable? By which we mean, given different data and different analysis, can we come to the same general conclusions? And I'm going to try to convince you that there's at least certain reasons to think that maybe we're not doing so well. This is from a meta-analysis, kind of a meta-analysis from a few years ago, from Mueller et al., from Simon Eichhoff's group, where they looked at they looked at 99 different studies of imaging studies of depression, basically case control studies comparing people with a diagnosis of depression to healthy people. And there were 3 previous meta-analyses, all of which had basically said, hey, look, there looks to be sort of consistent, findings across these studies. When they went back and sort of fixed some problems with the meta-analyses, basically what they found was that across these 99 studies, each of which had claimed to have found a difference between healthy and depressed people, there was nothing that converged across those 99 studies, no significant results. And that's consistent with some other data that's just recently come from this group in a preprint in a large case control study. And we think that one major aspect of this has to do with statistical power. We've known about the problems in statistical power in neuroscience from, since the Kate Button paper from 2013, which looked at both structural imaging and rodent studies and showed that, the majority of studies have very low statistical power, many of them down below 10%. A study with 10% power is basically a futile study, because even if there's a true effect, 9 times out of 10, you're not going to find it. And other work by Marcus Munifo and colleagues has shown that that this is actually a much more general phenomenon across biomedical research. Now you might say, well, that's fine. As long as we're controlling type 1 error, then if our power is low, that's fine. We just won't find things very often. But it turns out that that type 1 error may not be the thing that we're so worried about. And Ioannidis first pointed out in his 2005 paper, and Button et al. Laid it out in their paper, that one of the problems with low power is that it leads to a low positive predictive value. So this is basically the likelihood that if you get a positive result, that it's a true positive as opposed to a false positive. So it's conditionalizing over just the positive results rather than over all results, like the type 1 error does. And what they showed is that depending on your prior odds, even when you have low power, the likelihood of the finding being true can be very small because of that low power. And so this is consistent with something that Tagliarconi wrote back in 2010. Consistently running studies that are closer to 0% power than 80% power is a sure way to ensure a perpetual state of mixed findings and replication failures. And I think this is nowhere clearer than in the literature on a candidate gene association. So starting around 2000, people started publishing associations between various phenotypes and polymorphisms in specific genes this is an example, a meta-analysis of associations between polymorphism in the BDNF gene and hippocampal volume. What we see here is the effect size, the Cohen's d, which is the standard deviation effect size, by year of publication, and the size of the marker relates to the sample size. And what you see is early on, there were some very large effects reported in very small samples. Over time, you see what's come to be known as the decline effect, which is sort of a very general finding in science, as sort of effects, as sample sizes get bigger, effect sizes get smaller. And now we know from very large GWAS studies that none of these previously associated candidate genes are actually associated with hippocampal volume. And in general, the other thing we now know is that effect sizes for any particular genetic polymorphism are generally orders of magnitude smaller than this, generally accounting for less than 1% of the variance in any phenotype. So we wanted to ask a while ago, how well powered our FMRI study? So in 2017, we published an initial study of this, and I've updated this to data up through 2020 now. And basically on the left, we see sample size. This is log scaled, so it's actually better than it looks, right? This is the effect size that the study is powered to find, right? So your study needs to be powered to find a plausible effect size. If your study can only find effects that are as large as, say, the difference in height between a 2-year-old and an adult, that's not, you don't need statistics to find that kind of an effect, right? So the question is, for example, we can look in 2019, we find that the median study was powered to find an activation with about an effect size of 0.74 Cohen's d. The question is that a plausible effect size for FMRI? The problem is you can't look at reported effect sizes because they're basically kind of cherry picking results from a whole brain analysis, so they're going to be inflated. So in order to find realistic effect sizes, what we did was we took the Human Connectome Project data, did a group analysis for each of the tasks, and then fortunately, the HCP project had written down before the study what the regions of interest were for each task. So we took those, and then in order to be sure we were finding voxels where we were really certain that there should be activation for whatever the task is, we conjoined this with meta-analytic results from Neurosynth to basically try to find the voxels where we're really sure there should be activation, but that are independently identified from the actual data. Then we put those together to get an unbiased effect size estimate. So the question is, what are realistic effect sizes? And here we see on the left side, the bold % signal change. And most of you probably know that rarely would you expect for any fMRI task activation, rarely would you expect more than about 4% signal change on average. And that's what we're seeing here for the motor task, right? In the supplementary motor cortex, you see about 4%. In the precentral, you see about 3%. For other things like amygdala and the emotion task, you see almost 3%. And so this tells us that these are whopping effects, but the problem is that's not the effect size that we care about for statistical analysis. What we care about is the size of that effect with respect to the variability across people. That's what the Cohen's d quantifies. And if we look at the Cohen's d for these same tasks, we see that about the biggest Cohen's d we find are about 0.7, 0.8, and that's for the motor task in motor cortices or supplementary motor cortices, or for the amygdala in an emotion task. So what is this telling us? This is telling us that the median effect size that of all the studies published in 2019 that are in that database was that study was powered only to find the most whopping fMRI effects that you can ever hope to find, right? For anything interesting, those are going to be probably an order of magnitude smaller. So it's telling us that the literature remains badly powered. And if we actually take the sample sizes from the Mueller et al. Meta-analysis for the 93 studies reported there, we can see basically that over time, power has gotten better in the sense that the effect size that you're powered to find has gone down. Early on, you're only powered to find effect sizes of, well over 1.5. Now it's approaching 0.5. The median study from that meta-analysis was powered to find an effect a size of about 1 standard deviation. And just for reference, that's bigger than the difference in height between 13-year-olds and 18-year-olds, right? So that's, once again, kind of a whopping effect that you don't really even need statistics to compute. So, some of you may be familiar with this paper by Simmons et al. From a few years ago called False Positive Psychology, which laid out a bunch of issues in psychological research that they claim sort of were, driving irreproducibility. And one of the claims that they made was that, for any time a study reports 20 observations per cell or less, they need to provide a really compelling justification for that. And the argument is that in general, you would never plan to collect a sample of less than 20 per cell. And so when you do that, it's likely that you're basically using some kind of like flexible termination rule or interim data analysis, basically p-hacking to drive that small sample. And that's, an argument for, if we look at these studies in the Mueller et al., the meta-analysis, the majority of them had on this criterion a grossly insufficient N. We also know that small samples lead to high instability of statistical estimates. This is a demonstration from Felix Schonbrot a while ago. He has a subsequent paper on this, highlighting how correlations, if you have a true correlation of 0.25, your sample, the sampling distribution of the correlation, if you take samples of various sizes, you can see with small samples, it's all over the place. For example, from minus 0.25 up through 0.5, right? And it's not until your sample gets much larger, in this analysis over 160, until you reach some level of stability. We've seen this recently in the paper by Marek et al. That's on bioRxiv, which looked at this in the ABCD data set and showed that if you take small samples, you can get for the same phenotypes, the same variables, you can get wildly opposite findings simply due to sampling variability. And they show this, how this changes with sample size. And their analyses suggest that for brain behavior correlations, you really need samples upwards of 2,000 in order to reach that sort of point of stability. This also seems to be the case for predictive modeling. Gael Varkwa showed this in his nice cross-validation failure paper a few years ago, which showed, in general, for machine learning analyses, you also see this decline effect across a lot of different phenotypes. Okay, so the generalizability of neuroimaging results seems to be imperiled by low sample sizes. So how do we fix this? Well, one thing we can do is to work together, right? And I think the great example we've seen of this has been from the Enigma project, which I'm sure many of you are involved in, where a lot of people have come together to generate large data sets, that can allow us to ask questions with much more power than one could ask, within any particular group. And I think that's been really impactful. We've sort of gone another direction and tried to do this through data sharing. So we've developed this platform called Open Neuro, which openly shares a broad range of brain imaging data sets. But it's worth pointing out, it's easy to share data badly. If you haven't seen this little cartoon on YouTube at Data Sharing and Management Snafu, it's definitely worth watching. It's sort of painfully funny. This is a bit, the sad panda's asking another panda for the data. It says, I received the data when I opened it up, it was in hexadecimal. Yes, that is right. I cannot read hexadecimal. You asked for my data and I gave it to you. I've done what you asked. Is there a guide to the data anywhere? Yes, of course. It's the article published in Science, right? We all know that that's laughable. So we've been involved, for the last now about 7 years in developing a standard for sharing data that makes shared data really useful for people straight out of the box. And that's the Brain Imaging Data Structure, or BIDS, which basically provides both a file organization standard and a metadata standard for imaging data. And I'll just highlight, what a community effort this has been. It's now, it was started by Chris Gorgolewski and myself and a few others back in 2015. Chris moved on to Google back in 2018. or 2018, and it's sent. We were worried that it was going to die when he left, and it's actually since grown amazingly. We have an elected steering group that I'm part of. We have 6 maintainers who keep the project going. Over 70 contributors on GitHub. So it's really become, I think, an amazing success story in terms of like a community effort. So the idea behind BIDS is it takes these sort of uninterpretable data that you get off of a scanner in DICOM format, and it puts them into a format that looks like what most people would do in their labs given the choice. So we try to use standard formats. So for example, the imaging data are generally in NIfTY. The things like phenotypic data are usually in a tab delimited text file. When we need kind of dictionary-like formats, we use JSON. So we try to make it sort of, make the barrier to entry as low as possible. And then we provide, the standard provides both directory naming templates, like, how do you name your subject directories, and file naming templates. What are all the things that might go into a file? And these have gone through a ton of discussion to try to sort of work with the largest possible different number of data types. Now, one thing that's essential for a standard is to be able to know if a data set actually meets the standard. And so we worried about this very early and developed a validator, in part because one of the drivers for developing BIDS was the need to basically automatically ingest data into OpenNeuro without having a human curator look at the data. So we developed this validator. It's JavaScript. It runs in the browser, and so it can basically validate a data set before it ever gets uploaded, and tell you, is it valid or not, and it tries to give you some information about, how you might go about fixing any issues. This is what you see when you upload to OpenNeuro. One of the steps, the second step you go through, is a validation step where it basically checks your BIDS compliance of your data set, and if it's not compliant, you can't upload it. One of the great things about BIDS is the extension process that the community put in place. And we've now seen a bunch of different extensions, and there's a ton of ongoing work, particularly around kind of newer data types like process data, connectivity models, statistical models, image transforms, things like that. And so it's become a huge growing community effort. And it's been really widely adopted by, I would say that we don't have an exact number, but it's well over 40,000 participants' data that have been put into BIDS so far. All right, so let's move on to the next quadrant, which is robustness. Are neuroimaging results robust? And one way to ask that is, robustness basically means different analysis, same data, right? And this has become sort of general interest lately there's a number of different concepts that are used for it. The one that we sort of go with is multiverse analysis, the idea that, if you run a data set through many different plausible analyses, do you always get the same answer? And there's certainly work like this early work from Stegen that showed that, for some analyses, you can get very different results across different analyses. Ioannidis refers to this as vibration of effects. Now we know that there's a broad set of pipelines available to fMRI analysis. This is just within FSL. We were able to come up with 69,000 plausible analysis workflows simply by kind of combining all of these different choice points that are available within FSL, all of which are kind of things that nobody would look at each individual one probably and say, hey, that's obviously wrong. And we know that these ramify in the literature. Josh Karp did an analysis of what methods people report and saw that there were, across the 241 studies he looked at, there were nearly as many unique analysis pipelines as there were samples in the study. And Josh also showed in this well-known paper that these actually have an impact on results, right? So he took a data set from what was then OpenFMRI, ran it through almost 7,000 pipelines, and basically saw that you got wildly, even though you got a reasonable mean activation pattern, it was similar to what we had reported in the study, but in some places you could get 8 standard deviations different between the highest and the lowest statistical value being reported, showing that there is a substantial impact of the analytic variability. So we set out to sort of look at how this happens, how this works in the wild in a study we called NARPS, Neuroimaging Analysis, Replication, and Prediction Study. Again, I'm sure that there are some people on the call here who were actually co-authors on this. We had almost 200 co-authors on the paper that was published last year, really led by Tom Schoenberg and his student Rotem, who's now a postdoc with Tor Wager, along with a number of others, both from the economics world and the imaging world, because there's also a prediction market component that I'm not going to talk about here. So we collected fMRI data in Tel Aviv using a task that we've used before, designed to ask some particular questions about how people make decisions under risk. I'm not going to go into details of the task. We then recruited people to analyze the data via social media and conferences. And basically we said each team could have up to 3 people. They had to sign a non-disclosure agreement saying they wouldn't say what their results were until we gave them the okay. And everybody was offered co-authorship. That's why we have so many authors on the paper. We gave out the raw data set along with, if they wanted it, pre-processed data using FMRI Prep. And about half the teams used our pre-processed data. And we gave them about 3 months to basically give us yes/no answers back on 9 hypothesis tests, which were of the form, is there significant activation in some particular brain area for some particular aspect of the experimental design? We told them to use and basically analyze the data as if it was a study in your own lab, using your standard analysis workflow. The only restriction was that it needed to be a whole brain analysis. So we got results back of the 84 teams that took the data. We got results back from 70. And in addition to their yes/no decisions, they also uploaded whole brain statistical maps both before and after thresholding. So interestingly, we asked them to give us a written description of their analysis workflow based on this Kobados report from a few years ago. And none of the 2 teams used an identical workflow. And strikingly, it was often also hard to tell what they had actually done, even from these 10 to 15 page long reports. So here's sort of the main finding. On the y-axis, you're seeing the proportion of teams basically that said yes for each of those hypotheses. And the hypotheses are sorted from sort of lowest number of teams saying yes to highest number of teams. So just to be clear, if everybody agreed, these numbers would all be either 0 or 1, where 0 is there's no activation, 1 is there's activation. What we see is, there's 3 hypotheses, which were all sort of, between subject hypotheses, I think all about the amygdala, if I remember correctly, where everybody agrees pretty much that there's nothing. There's 1 hypothesis where pretty much everybody agrees that there's activation. And then there's these 5 in the middle that are from 20 to 40% of teams saying there's activation. So that's a pretty shocking degree of irreproducibility. Another way to look at this is that there were, across the teams, for those 9 hypotheses, there were 33 different patterns of outcomes. If we look at the whole brain results, taking the thresholded maps, we see that, that there certainly is good overlap, but the highest degree of overlap, the voxel with the highest degree of agreement, only has 76% of teams agreeing, right? So even for the places where there's pretty clear agreement, there's a lot of variability. Now, interestingly, when we looked, this is a correlation matrix heat map of the whole brain statistical maps, the Z statistics. And what we see is that there's one group of teams that are pretty highly correlated with one another. But even for teams that are really highly correlated, the red and the green at the top basically is telling us, does the team report a positive or a negative outcome for the hypothesis test that this is testing? And you can see that even for teams with really highly correlated maps, you can get differences in the yes/no results, suggesting that the thresholding operation is certainly part of the answer, though we did some analyses that suggest it's not all the answer. We also had a number of teams, 8 teams here, whose maps were anti-correlated with the rest of the teams. And when we dug in more deeply, we found that they had basically misspecified the statistical model. Happy to say more about that in the discussion. But nonetheless, if we do meta-analysis across those groups, we do find reliable findings. So for example, for the analysis I was showing earlier, we find really robust, this is that hypothesis 5 where everybody pretty much agreed. And the meta-analysis across the image-based results, the statistical images, shows us, good agreement. So it's pretty clear that the robustness of neuroimaging results is imperiled by this extreme analytic flexibility. So I'm going to talk about a couple of different things that we think are important. One is pre-registration, which is really registering the sample size in the analysis plan up front. Now, many people worry, and I used to worry, that this is going to prevent you from doing any kind of exploration. That's not what it does. What it does is it makes sure that whatever you do that's exploratory is sort of clearly distinguished from whatever analyses were planned. And you can, every paper we write ends up having a section that says, well, we pre-registered this, but we had to do some things differently. As long as you're clear about that, it's never been a problem for us. The effects of pre-registration in biomedical research have been pretty striking. In the year 2000, the NHLBI, which funds cardiovascular trials, started requiring registration of the primary outcomes of clinical trials. And what we see was before that, this is from Kaplan and Irvin's paper in 2015, before 2000, each of these with a plus sign is a positive reported result. So the majority of studies claimed that their treatment was beneficial. None of them claimed that it was harmful, which is what above 1 means. Whereas once those outcomes had to be registered, now nearly every study finds a null effect. A couple find a positive, one finds a significant negative effect, harmful effect. So this shows that, it isn't as if medicines just stopped working in 2000, right? People stopped being able to make it look like their medicines worked by sort of fudging the primary outcomes. Another thing we think is important is to move away from using a single analysis workflow and towards multiple analysis workflows. And so we've been doing work on a project called FitLens that basically allows you to take a machine readable specification of the statistical model from this thing called the BID stats model specification and implement that using, currently we can implement it using either NiLearn or AFNI's statistical modeling tools. And so this allows you to sort of easily generate and run a bunch of different statistical models that you can then think about doing meta-analysis across. The other thing that we think is really important is moving towards using well-tested software tools. I mean, we all know that in each of our labs there's software that was written by a postdoc 8 years ago that people have been sort of making continue to work, and nobody really knows how it works anymore, but it all kind of has duct tape around parts of it. And so that's just a recipe for disaster when it comes to software. And so we've been working with a bunch of other teams to develop a framework that we call Knifepipe, which is an extension of the kind of outgrowth from the FMRI Prep tool that we published a few years ago, which basically includes a bunch of sort of software infrastructure and middleware that supports the ability to build really robust and well-tested software tools. And one of the ideas that we developed back in BIDS, when we were first developing BIDS, was this idea of a BIDS app, right? Which is basically a containerized software application. I'll say more about containers later. But the idea is that the container encapsulates all the software and all the dependencies, allowing you to better reproduce the analysis. One example of a BIDS app that we've developed in this NIPREPS environment is this thing called RIQC, which provides a tool to very easily generate a group quality control report along with individual reports for both structural, T1 weighted structural MR and functional MR. Another one that I think is sort of the flagship of NIPREPS right now is FMRI Prep, which has gotten a ton of attention in the last couple of years. It's basically meant to be a kind of a robust end-to-end fMRI pre-processing toolkit. Does processes both on the surface and in the volume, and it tries to basically use all the data that are there to do the best possible job of pre-processing. This is just an example. It uses Knifepipe, which generates these kind of workflow diagrams. And one of the things that Oscar Esteban and others in this project have done is developed a number of nice tools for visualizing the results, right? You don't want to give people a really powerful workflow and say, hey, go use this and just trust the results. So we try to basically figure out where do people need to look at the results to make sure they worked right. And clearly, in this example, EPI to T1 weighted registration is really important. And this particular tool provides a really easy way to sort of visualize that. I don't unfortunately have data from 2021, but at the end, this is data from the end of 2020, beginning of 2021, where we saw more than 100,000 successful runs in 2020. The software has a telemetry system built in where it's opt out. So if you want to turn it off, you can, but if you don't, then it basically, whenever it runs, if it runs successfully, it sends us a report saying, hey, it ran successfully. If it fails, it sends us a report saying it failed and what was the error. And so we can see that it's getting a ton of usage, sometimes over 12,000 runs in a week. Okay, so let's move on to replicability. Same analysis, different data. And we've known for a while that there are, potential problems in the replicability of fMRI data. This was an early finding from Fowler-Buchel and colleagues, which showed that a bunch of brain behavior correlations were not replicable. Now, one challenge is that, what does it mean to do the same analysis? Even if you describe your analysis using this sort of standards compliant approach, it's often hard to tell what was done in an analysis. And so we think that code sharing is, essential to actually know what a person has done. And I think this is captured well in this quote from David Donahoe. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment, code, and data that produce the result. Another question is, what do we even mean by the same result in fMRI? And Torwager and Chani Wu had a paper a while ago that I thought did a nice job of laying out how, you often see basically people saying that they replicated a finding when in reality, if you actually look at the results, they don't seem to really overlap at all. Almost half of the replicated findings had peak coordinates more than 15 millimeters away from the original findings. And so, we think that replicability is imperiled both by a lack of code sharing and by flexibility in the definition of replication. So clearly, we need to share our code, right? And many people are worried about sharing their code. And I think, one thing to ask yourself is if your code is not good enough to share, then why do you think it's good enough to share the results that rely upon the code in a publication? So, there's a lot of different approaches to sharing code. We like sharing, obviously you can just put up a GitHub repo. There's a number of reasons why that's not a best practice. And so we like sharing code by giving a snapshot of the code and actually assigning a DOI to it using the link between GitHub and Zenodo. So this is from that NARPS paper. You can see we have fully reproducible code available at this DOI. If you follow that link, you're taken to Zenodo and you can grab exactly the code, the specific release of the code, version 2.0.3, that was used to generate all of the figures in the paper. We've seen also that, the sharing of code has an important impact on reproducibility more generally. So we had published a preprint last year that basically, analyzed the ABCD data set and showed some issues with the stop signal data, the behavioral data in that study. And this is a piece of text from the paper. We also made all the code available when we published the preprint, and the data are already available through the NIMH data archive. So the Sage Han from the ABCD team tried to rerun, kind of looked at all of our analyses and realized that we'd made an error in our code. Basically, we were doing some indexing and we had this really ugly, nested Boolean thing going on, and basically just had a parenthesis in the wrong place. And so when we reran it after fixing that, he surfaced the error. We thanked him and reran everything, and it changed the results, significantly. The overall, story in the paper didn't change, and the paper went on to be published in eLife. But it shows how sharing code and sharing data can really help make your work more reproducible, and and it also shows that when people find a problem with your code, that's not necessarily like a personal attack, right? That everybody wants, one would hope in science, everybody wants to find the right answer, and what you should do is take that feedback and work with it. Okay, finally, let's ask about reproducibility. Same analysis, same data, right? It seems like this should be pretty easy. And so we know that the sharing of both data and code are necessary to ensure that. The question is whether they're sufficient. And so here's an example, not from an imaging study, but from an analysis I did a few years ago, where some of you may have heard of this myConnectome study, where I collected a ton of data on myself, including imaging data and RNA-seq from white blood cells, and did a bunch of analyses about how things changed over time. And this was kind of the first study the first paper I ever published where we released a quote unquote reproducible workflow. And we did that by releasing a virtual machine, or a definition of a virtual machine. So Vagrant is a tool to generate a virtual machine from a definition, this Vagrant file. And you basically say, hey, Vagrant, go set this up. It pulls all the dependencies, it pulls all the code, and it goes and runs everything. So it seems like that should be sufficient to give us reproducible analyses. I got an email a couple years later saying, hey, I'm an undergrad, I'm working with the RNA-seq data. I'm having some trouble reproducing the RNA-seq results using this WGCNA thing. There's supposed to be 63 co-expression modules, as in the paper website and supplement. But when I run the scripts, I only get 60. I also reran the analysis with the VM, and the result is the same 60 modules. Essentially, the same code is run on the same data, but arrives at 2 different results. What's going on? And this person noted, I noticed that the WGCNAR package has been updated a number of times. Maybe this is causing the issue. And that's exactly what it was. So we had run the analysis. Fortunately, it's always best practice to put the specific version of any software package you use into your paper. We had noted we used version 1.7 of that package. Subsequently, it was updated to 1.51. But the Vagrant file that specified how to create the virtual machine didn't specify an exact version of this package. It turns out that within R, at least back then, there weren't great tools for specifying particular versions of packages for code. It's gotten easier now. We also, and so that's one, clear differences between software versions are kind of an obvious version, but there's also, even when the software remains the same, exactly the same, you can see differences in results. So this is some work from a few years ago by Tristan Gletard and colleagues, which basically showed that, the same software build of FreeSurfer run between different Linux versions can give you not hugely, but, appreciably different estimates of cortical thickness. And similarly, same Linux version, different software builds, you can get appreciable differences in cortical thickness estimates. So clearly, being able to run the same analysis on the same platform is really important. So how do we do that? Well, one way to go about doing it is through using containers. So I mentioned this before. A container is basically sort of like a really lightweight virtual machine. It includes basically the specific versions of all software libraries, except for the like really deep, basically the kernel of the operating system. And so the software that's most popular for this is called Docker. It's basically a tool that can run on, on Windows, on Mac, on Linux. Underneath the hood, it's running a Linux kernel. And so it's basically like being able to run your code using all the same versions of all the same libraries and everything on a Linux kernel, depending on, regardless of whether you're on a Windows machine or a Mac or an actual Linux machine with maybe a different version of Linux. So it allows you to have much more control over the exact platform that the software is running on. The other thing you can do is you can share an image of that full container, so that somebody else can fire it up and have exactly the same system you're running on. So we've, for example, in FMRI Prep, the way that we recommend people to run FMRI Prep is actually using Docker. So this is the command that you would need. If you want to run FMRI Prep, all you have to do is install Docker, which is easy to install on any system, and then just run this one command from the command line. And this is basically telling it where to find the data, where to put the outputs, the version of FMRI Prep to use. It pulls from this Poldrack Lab site on the what's called Docker Hub, tells it what to do, where you're going to put the outputs, and so this is a very easy way without having to go install all the dependencies of FMRI Prep. They're all contained in that image that gets pulled down by Docker and run. So now you can rerun FMRI Prep 20.2.0 and modulus in places where random seeds might come into play. You can pretty much get the exact same outputs. And it is true that random seeds actually do have an effect here in some cases. One of the things that what we'd like to do is have ultimately kind of a fully automated way to go from code and data to results. And so we did this in the NARP study, where we basically used something called continuous integration, which is a sort of technology that's often used in software engineering, basically for continuous testing of code. So what we did was we have our data in this repo on GitHub. Sorry, we have our code in this repo on GitHub. We have our data in Zenodo, we have the Docker image that's in Docker Hub. And whenever we push a new version of the code to GitHub, it automatically triggers this system called Circle CI to basically pull the latest code, the latest data, the latest Docker image, and basically run everything. You have to basically define all of your analyses as tests, and it will go run all those tests. And then it will basically, if it fails, if something crashes, it'll tell you that. If it succeeds, it'll say, hey, success. And then the results are all saved out as an artifact on Circle CI, and you can just pull those down. And so if you want to go do this, you can do it, and you'll basically get all the same results that are reported in the paper. Okay, so just to kind of, end up and sort of, review sort of, where we have ended up. We think that there's clearly issues with each of these aspects of reproducibility, but we also know how to fix them, right? We know that, to make things generalizable, we need larger samples, we need consortia like Enigma, we need data sharing like Open Neuro. To make things more robust, we need things like multiverse analysis, we need pre-registration, we need really robust and well-tested software tools. To fix replicability, we need code sharing and platform sharing probably. And then even for same data, same analysis, we definitely need data and code sharing and also platform sharing through technologies like containerization. So just want to end by thanking my group who have looked like this for the last couple of years. Ton of people who've been really great collaborators. And just mention, we've started a new center at Stanford as part of Stanford Data Science now called CORES, Center for Open and Reproducible Science. And we're starting to develop content there. If you're interested, have a look at our website, and you can get on our mailing list and see when we have upcoming talks. And I will stop there and be happy to take questions.
Thanks, Russ. So if you have any questions, please post them to either the chat or the Q&A. Thomas, are you saying you would like to be unmuted? I guess. Yep. Okay. So Armin, could you unmute Thomas? He's got one question.
Hi, Dr. Poldrak. So 2 questions, if I may. First of all, given like the issues we're seeing with reproducibility relative to the effect size, if we're trying to address power by having larger consortia, aren't we eventually going to reach a point where just about anything would be significant with a large N?
Yep, that's a huge issue, and I think it's becoming clear, especially, it was clear even many years ago in this paper by Peter Bannattini's group, Javier Gonzalez Castillo, where he collected a ton of data on on individuals on tasks, and basically saw that with enough data, pretty much every voxel in the brain shows a significant task response, like 90-something%. And now what we're seeing with UK Biobank, where you have tens of thousands of people worth of fMRI data, you're exactly right that any incredibly tiny effect becomes significant, and this is an issue that's people have known about for a long time with null hypothesis testing. So I think what it suggests is that we need to move beyond null hypothesis testing, for our analyses, and move towards tests that are more focused on things like effect sizes, predictive accuracy, that sort of stuff. I think, yeah, so I agree. It's a huge problem, and it suggests we need to move away. We've been, in some ways, the low power has kept us in this regime where like there's some significant stuff, but not everything is significant. And now we're moving out of that regime where that has worked.
So you'd support something like, say, equivalence testing over standard null hypothesis?
Well, I think that might be. I mean, that might be part of the solution, right? That's still a kind of a frequentist approach. I guess I'm more intrinsically pulled towards Bayesian approaches, but I think that there's room for all. I mean, there's room for confidence intervals, there's room for equivalence testing, there's room for Bayesian analysis. I think that all of these things provide some sort of. I'm not like religious about this sort of stuff. I think all of these things provide some way to address the problem, and I think we just need better tools to do all of them.
All right, and another, if I may, but when we start looking at these large consortia and we start doing like machine learning tasks or classification tasks or something like that, we're running the data multiple times on the same data. Some groups may be publishing using the same data multiple times. And we get in, how would we address, say, like, you've run the FBurn data set and done 30 publications on it. Are you at any point going to do a POCOC correction or something of that nature on it? Or in worst case scenario, you have, like, say, a 3% difference in accuracy between the two winning classification models, and to validate that, you need a data set on the order of the millions.
Yeah. I think this is another great question, and it's something we worry a lot about. We actually had a paper in eLife last year, a couple years ago. I just posted the link in the chat. We called it data set decay, right? The idea that over time, you're going to get this increased number of false positives unless you're controlling for the number of studies being done, a number of analyses being done on the data, right? If you have any kind of publication bias, right, where only the positive results are getting published, then over time you're going to get this proliferation of false positives. And so I don't know what there are ways to think about this, right? So you could imagine, certainly one approach which hasn't been taken by any of the big consortia is, basically only sharing a portion of the data and then having, some way for people to upload predictions on test data, sort of like what people do in machine learning contests, right? So use that sort of approach, but have kind of like, a hidden data set, or use some sort of like, differential privacy sort of approach. So, people can get different versions of the data. I think there's various ways to think about addressing that, but that's really a societal kind of, incentive problem, right? Nobody who's collecting these big data sets is incentivized to sort of address that problem. So I see Carlos has a question. There's several efforts to have open data, like Stanford AMI, shared data sets, International Data Portal. Are there efforts to integrate those efforts into a consolidated platform or standard? The short answer is I don't think so. And I'm not sure that that's necessarily the right way to go. I mean, in part because I think there's different data sharing approaches and, different sort of focuses. And I think that having one I think we actually see the problems with having a single platform in the NIMH data archive, right? Where it becomes really challenging to handle this platform that's been built to be able to share this really broad set of things. Whereas if you go to Open Neuro, right, it's built to share one kind of data, which are data that meet the bid standard, right? It's a single standard, and so there's only a limited type of data, but the data are really, sort of easily accessible and clearly organized. And, I'm not sure it makes sense for us to start sharing like genomic data, right? Even if there was another validator, because, there's also something to be said for sort of domain-specific repositories where people in the field know to go there to get data, right? You can go to a place like, one of these sort of domain general data sharing projects like Dryad or, Zenodo or something, and you'll find FMRI data there, but they're really hard to find, and they're not necessarily going to be organized in a way that's useful, right? Whereas if you go to Open Neuro, for example, the data there are shared in a way that makes them really useful for people because they all meet the standard. So I'm a little bit more of the let lots of flowers bloom type of approach, so that we can have sort of like, for people who want a rose, they can have a rose. For somebody who wants a camellia, they can have a camellia. There you have all the different types of flowers that somebody can go pick from.
I have one question, which is you touched on it. I think you mentioned briefly random seed, right, at one point, and so I think this is a huge issue, and kind of especially like with machine learning approaches, and also FreeSurfer, right? So that paper that looked at OS FreeSurfer, those results I think are in large part driven by differences in random seeds initializations in different operating systems, right? Or versions of operating systems. So and there are strategies for trying to deal with that within particular analyses in the machine learning community, but I think there hasn't been a lot of systematic, in the neuroimaging world, systematic approaches to deal with that. So I think that, I don't know what your thoughts are on, or what sorts of things you think are trying to, address this. But to me, it seems like that could be the source of a lot of variability. So like if you're using free surfer results, for example, to do classification, right? And someone else takes the same data and does the same classification using the same pipeline, they might get a different answer, right? So it's those kinds of things, especially when you start to see, can we make something useful about these approaches that might have an impact on a person, right? So then it becomes a huge, even greater issue. So just more of a comment, I guess, but I wanted to get your thoughts on that.
I think it's a really important. I was trying to find there's a preprint that recently came out from Mike Millam's group that we've been involved in that was addressing some of these issues around, basically, so we know that in FMRI Prep, there are some issues around random seeds. We've started now, being able to at least, I don't think we have full control over random seeds, but we can at least, we store them out for a particular run. But Mike Millam's group was basically trying to set up, so they have this thing called CPAC, right? This kind of, pipeline, really highly configurable pipeline. And they wanted to basically see, can we replicate the results of FMRI Prep as exactly as possible using CPAC, right? And a bunch of work went back and forth, and ultimately they were actually able to do it with a pretty high level of reliability, at least in terms of like, if the question is like, how similar are the functional connectomes that get estimated, from the pipelines. So I think that shows that it is possible to harmonize pipelines across different packages. Obviously there's some common software components there.
Mm-hmm. That was by Jinhui Li, right? That was that paper, I think, where they did pipelines, multiple options within pipelines. Yeah. So she's actually a student of mine now at Georgia, at Georgia Tech. So, cool. But I think that's a slightly different issue. It's an important one as well. But I'm thinking like of iterative approaches, right? So if you're running multiple solutions multiple times, and and there's no closed form solution, right? So then in that case, you could run it once. You could take the average. You could do some other statistical approaches to make it more robust, or more like probable that someone else running it would get a similar answer. Again, a simple example of this is what we do with the GIF software in ICA. We do this Acaso approach, right? So you just kind of it's not perfect, but the idea is you get a slightly different ICA solution every time. And if you run it multiple times and take the sort of the best solution, finding what you mean by best, then someone else runs it, they might get something similar. So
so I agree that there's two things we need to do, right? One, we need to know where is it that the random seeds actually have a significant effect. And then we need to do, as you're saying, and make sure that we're assessing the robustness of the results to that particular, for that particular portion of the analysis.
And then let me just see if I've got another question, but I want to ask if other people have questions.
Looks like there's some.
Okay, so I'll jump in again, which is the issue of, so I think it's really valuable to try to get robust results across multiple pipelines, right? I think that's very valuable, but I also think, sort of counter to that is there's a reason why people choose particular pipelines, right? And sometimes they're subjective reasons.
I think those reasons are rarely good, right? They're usually like, somebody else in my lab was using that pipeline when I showed up, or the postdoc who knew something about that method brought that pipeline to our lab. That's, in my experience, that's where pipelines come from. It's not like
I think there's a lot of that, but I think there's a lot of good reasons as well, right? So there are well-justified rationales for certain choices, right? And there are lots of papers on this, right?
I agree that
you could certainly say, let's just look at the average, right? But then the question is, maybe there's, maybe there is a set of options, and I think Jinhui's paper got at this a little bit. Maybe there's a set of options that are more important or give you more likely optimal solutions given a question that you're asking, right? And so I think there's a lot of room for that, but that hasn't really been explored too much yet. But it's kind of a second order question, I think.
Yeah, I agree.
Okay,
so it looks like there's some questions in the Q&A.
I see.
Our NIH friends are here.
Right. So Peter, I'll just read it out. I guess ultimately one goal is the generation of biomarkers. Is there hope that once we find a highly robust result that seems to have predictive value, what is the hope and procedure for then attempting to scan individuals clinically using these functional templates? Is there enough stability to go back and draw inferences about individuals?
I think
one of Peter's favorite questions.
Yeah, right. I think that's a really critical question, and I think we don't know. I mean, I think that, what we've clearly seen from, for example, these ABCD analyses is that, for kind of gross types of things, it's not clear to me that there's going to be enough predictive power of FMRI to make sort of strong inferences about individuals. There may be corner cases or, particular disorders where it might be true. Certainly I think, what's coming out in sort of larger studies of a number of different psychiatric disorders right now is that, that they're seen when the samples are big enough, the ability to, make strong predictions, even if it's just like diagnostic classification from FMRI, is pretty weak. Now that probably is saying as much about the diagnostic procedure or the diagnostic classification itself as it is about FMRI. But nonetheless, I think
weak. What's weak?
Weak. Well, weak is either none or let's say that you can predict with like 60% accuracy, without. And one challenge, right, to all of this stuff is that we pointed this out in a paper a few years ago in JAMA Psychiatry that, we studies that are like doing case control studies of, say, schizophrenia, right? They're generally collecting equal size samples of people diagnosed with schizophrenia and healthy people, right? But the base rate of schizophrenia is an order of magnitude smaller than that, right? And so any sort of classification accuracy estimates that we get from a case control study like that,
probably 2 orders of magnitude.
Yeah, probably 2 orders of magnitude. So any estimates of classification accuracy we get are going to be really overestimating or underestimating the false negatives, or sorry, the false positives, right? Because of Bayes' theorem, right? We know that if you have something that's very rare, most positive test results are going to actually be false positives, right? And the higher, holding all else constant, the higher the base rate, the more likely something is to be a true positive. And so I worry a lot about the generalizing from the kinds of results that we get in these studies back to specific individual results. And I think there's a whole literature that I've seen a little bit of, but don't know enough of there was a group in the MacArthur Law Neuroscience Project called the Group to Individual Inference Group, which is all about exactly this question. This was in the context of sort of like, the law, right? Can we, if we know something about lie detection, how in groups, how do we, what can we say about this particular person, whether they're lying or not in this particular case, right? And so there's some interesting work in that area, around, sort of like the statistics of that kind of inference that I think we could probably learn more from. I don't know it well enough to say much, but I know that there's a lot of work there.
Yeah, no, that's a great point. And I think ultimately the where it's going to be, I guess what is weak is probably going to be dependent on the question, right? Because with a clinical question, right, it'll depend on the population that you're interested in distinguishing and what the prevalences are and all of that. So
yeah.
I'm a little more optimistic about the schizophrenia one because we've looked at obviously a lot of data, but we're looking across many, many different data sets now, and we're getting very robust, patterns of differences that in and of itself isn't necessarily going to be a useful, clinical question, but it's an important, if you can't solve that one, right, you're not going to be able to solve more.
Yeah.
So let me jump to Sean's question. I'd be interested in your thoughts about efforts to improve reproducibility with better data acquisition using denoising, such as multi-echo, as well as ultra high field, higher resolution studies, more extensive sampling from individual brains. And he mentions good arguments in extensive sampling for complete models of individual brains by Nasaleris et al. Or precision fMRI paradigms. Maybe.
Yeah. Thanks, Sean. I think that's another great question. And I'm all in on a number of these things. Certainly, I think, better, so we've moved to using multi-echo now because I think that the ability to do that sort of denoising is really important. If we had a good, high field system here that we could do human imaging on, we'd be doing that. And we've certainly, my lab has moved, in the new studies we're doing now, we've moved exclusively to doing basically extensive sampling from individuals. So we're not doing like, scan somebody for an hour anymore, for exactly the reasons that are laid out in papers like, the stuff by Thomas and Kendrick and others. There's a whole recent special issue of, actually I have it right here, Current Opinion in Behavioral Sciences on Deep Imaging Personalized Neuroscience, which has a bunch of papers. In fact, that paper by Nasaleris et al. Might be in here, but I think, or at least they have a review in here. So I think that moving towards that sort of approach is really important for getting more precise estimates and allowing us to sort of, go beyond the kind of the very gross kind of, lumping together of people based on registration, up to an atlas. Obviously, when it comes to asking questions about individual differences, we still need large samples, right? So there's no getting around the fact that if you want to get robust estimates of either group differences or correlations, you need large samples. And so the question is, how do we bring these together? And obviously there's really challenging issues around, like, if you want to study, sick people, how do you get them to come back and scan for 10 hours if that's what you need to get like really precise estimates of what's going on? We're just about to start trying to do some dense phenotyping on people with obsessive compulsive disorder. And that might be one of the easier, psychiatric disorders to work with in this context. Doing it, for example, with people with depression is going to be really challenging. And you're going to run into, another thing actually that is, I think, a problem in psychiatric imaging in general are the selection processes that are involved, right? So, we think of like, when we bring somebody in to scan and they have a diagnosis of depression, they are depressed, right? We put them in the depressed pile, but they're not a random sample of depressed people, right? Because a lot of depressed people are so depressed, they would never show up for the scan, right? So there's already a huge selection effect going on there. And if you think about it longitudinally, right, there are going to be days when the depressed person just can't get out of bed to come to the imaging center. So we're going to also have a within subject selection effect. And I think those are really challenging for these, for these sorts of things. And I guess, I have selection effects are much more on my mind just because of thinking a lot more about collider bias in general, and the way in which, like, we can get, if we're trying to understand relationships between variables, but we have selection on those variables, we can actually mislead ourselves about, what the ultimate causal relations are between those things. You can get, you can basically even get, correlations flipping in sign because of selection effects. There's a great paper by Rettelmeyer and someone else recently on kind of helmet wearing and hospitalization, which shows exactly this. So I think that there's a lot of things there to worry about, but nonetheless, I think that dense phenotyping approach is exactly the way to go.
Just a comment too, in terms of just scanning and duration of scanning. There's a postdoc here, Marlena Duda, who's been doing some work with spatially constrained ICA, and she's been able to show in some pretty large data sets that you get robust group differences with as little as 2 minutes of resting fMRI data, which is kind of the polar opposite of the sort of, you need 30 minutes, right, of data to get a good result. But it appears that in some cases maybe you can regularize the data if you have a focus.
I mean, well, if your prior is strong enough, you're going to have perfect reliability, right?
Yeah, but we're looking at group differences here, not within subject comparisons to a template. So this is a classification, cross-validated.
Got it. Cool.
And it's well above 80. It's around 80%, I think, or so.
Cool.
One more question from Chris. On an institutional level, do you think the publishing system itself can change to address, so we should stop there. Yes or no? No. To address these problems in neuroimaging and other fields. For example, how has requiring pre-registration reduced p-hacking?
Yeah. I think it has to change. You know, it's really a question of what the incentives are for, you know, at a lot of different levels, right? At an institutional level, you know, how do you get people to stop counting numbers of Science and Cell and Nature papers when they're, you know, looking at whether somebody should get tenure, right? Or, you know, even number of papers, right? Or impact factor, all that sort of stuff, right? How do we make it such that, you know, our assessment of individual scholarship focuses more on the quality of the work and the quality of the questions that are being asked. I think we've lived in this world for so long, right, where it's like the paper is necessarily a positive result, and like people are disappointed if they find a null result, right? Because it's like, I really wanted this to be true, right? When in reality, it's the goal of science is to ask questions, and questions that have a no answer are just as important as questions that have a yes answer, but it's like we're hiding all the no answers, right? And so we have to keep asking the same questions again. And so I think it's fundamentally important for us to change how we do it. I don't know how to do it. The problem is that humans are really good at gaming pretty much any system you build. And so my worry is, you know, it's like John Ioannidis has this great paper called like the fragile vase of science, where he makes this point that, like, you know, there's all these people with ideas about how we could change the publication system, how we could change other sorts of systems, but those are all just hypotheses. And right. And without the ability to do like randomized controlled trials to see, like, you know, is this a system that would give us more reproducible literature than this one? And we obviously can't do that. Then it's really hard to know, you know, whether we're just trading one bad system for another kind of shinier, newer bad system. And so it's hard for me to know exactly what the right way to go is, but it definitely seems like we have to do something.
Seems there is the field's starting to become more aware of it,
Yep.
which is the first step.
Yep.
And there are some efforts, you know, like Aperture and you know, which is an OHBM open publishing venue. Question from Pierre Belloc regarding computation. The infrastructure you mentioned with Docker and FMRI Prep has computational cost, and this in and of itself can be a barrier to reproducibility. Can we reduce the cost of fMRI analysis? Is that something that's important?
It's a good question. I mean, it certainly is important, you know, both in terms of like, you know, barriers, especially as you start talking about like, you know, the types of machine learning, deep learning sort of stuff that, you know, only DeepMind and Facebook have the computational resources to run, right? That's like that's the end game of that, you know, that kind of like race. And obviously we don't want to end up there, because then you just can't reproduce anything unless you work for one of those two companies or some other company that has a ton of money. So, and there's also, you know, climate cost, right? It's like, you know, when we're sitting here, you know, using all these cycles on machines, we are, you know, we're creating carbon, right? Or at least we're not creating carbon, we're releasing carbon, we're causing the release of carbon into the environment. And all those things are important, I think. The problem is that, you know, that type of work is hard. Like the optimization in terms of like, you know, computational efficiency is hard. It's not something that, you know, most people in our field are trained to do, and it's not going to get you fancy papers, right? So like we could probably like we could spend a year and take FMRI Prep and make it run much more computationally efficiently. But, you know, nobody is going to like give anyone tenure for doing that, right? They're going to give someone tenure for coming up with a fancy new method or coming up with a fancy new result. And so I think there's a lot of issues about just like, what are the incentives to do that? And especially when things are moving, like, you know, we do that, and then, you know, 5 years later, nobody wants to use FMRI Prep anymore. They're using some other new toolbox that won't be optimized. Now, the one thing to say is, like, to the degree that we can share optimized components, like optimized middleware or infrastructure components, I think that's one way to sort of try to address this problem. There's a group that we're involved in that's really being kind of led by Damien Fair and Mike Millam, called InMind, which is an acronym for something like, never mind, this neuroimaging method is not duplicated. The idea being, like, a lot of us are working on similar sorts of stuff, right? And we have people in our labs, like, all building basically software tools to do the same thing, right? Why can't we share those infrastructural tools? We can all still end up doing our different high-level things and get our papers out of those. But the idea is, like, we should have well-tested, just like, you know, a lot of us, for example, use scikit-learn right now, right? That's a really well-tested piece of infrastructure for doing machine learning in Python. If it would be nice to have well-tested infrastructure for doing a lot of other stuff that we do in neuroimaging that we could all share. And then that would, you know, those kind of things could be optimized, right? And then some work could be, it would make more sense to put work into that, because then a lot of people would be using it, and that would be, you know, sort of, that would have a lot more impact on the community.
Great. Well, thank you. It's 12:15, so I want to, I know you've got to go teach a class, so I wanted to be sensitive to your time. This will be continued, I guess, because we're going to have the recording will be released, and there'll be a Slack channel created. So there may be some questions we'll throw in there that that we didn't get to, or if others have follow-up questions, we're hoping that, yeah, you'll be able to engage with folks there, and maybe we can have some you know, sort of flesh out some of these interesting questions a little bit further. So thanks so much. It's been great. It was really, really interesting, and a lot going on, and a lot to think about. And we really appreciate your time today. And with that, I guess we will close this session of the BrainSpace Initiative, and let Russ get to his class. So.
All right. Thanks, y'all. Thanks for the invite. Good to see you.
Bye-bye.